Okay, well, welcome. So my name is Professor Julian Savulescu. I'm the Uehiro Professor of Practical Ethics and a co-director of the Oxford-Martin Program on Collective Responsibility for Infectious Disease. Normally we wait till 5 minutes past, but since there's no more chairs left, we may as well get started now. Welcome to tonight's discussion on the gene editing revolution, should there be limits. We're a part of an enormous biological revolution today. 3 great events have occurred. The creation of induced pluripotent stem cells, whole genome analysis and gene editing. And tonight we have the opportunity to discuss gene editing. It's a great honour for me to welcome really 4 of the world's foremost experts in this area. Dr. Andy Greenfield, who is second from the right, as you see the panel. There's a seat up the front here. Is the program leader for the Medical Research Council at the Harwell Institute. Closest to me is Professor Robin Lovell-Badge, the group leader and head of Division of Stem Cell Biology and Developmental Genetics at the Francis Crick Institute. Second from the end is Professor Alison Murdoch, Professor of Reproductive Medicine and a gynaecologist at the Newcastle Fertility Centre at Newcastle University. And at the far end is Dr. Gyngell, a member of our centre and a Marie Curie Research Fellow working on gene editing. So we will proceed by Chris summarising the technology for around 10 minutes, and each of the discussants will give a 5 to 10 minute perspective, and then there'll be an open discussion. So there will be almost an hour for you to air your thoughts. I have a couple of housekeeping announcements. One is that this is being recorded, so if you don't want your question recorded, if you could let Clara know at the end and we will delete it. This is the first event in the Hilary Term lecture series, New Frontiers, Pushing the Boundaries of Science and Technology. So the Martin School website will describe the other titles, but they are Thank You for Being Late, An Optimist's Guide to Thriving in the Age of Acceleration. Is a Post-Truth World a Post-Expert World? And Expanding Frontiers in Space Exploration. For those of you who engage in Twitter, if you want to tweet, the hashtag is hashtag OMS Frontiers. So without further ado, I'd like to turn over to Dr. Gyngell to give us a priming background.
Thanks, Julian, and thank you to the Oxford-Martin School for hosting this event and inviting me to speak. So I've just been asked to give you guys a bit of a background to the technology and some of the ethical issues that this technology is raising. So we've all heard about genetic engineering, and we can think of genetic engineering as a broad term that can apply to any technology that allows us to alter DNA sequences. And for decades, we've been able to use quite crude genetic engineering technologies to alter the genomes of other species of animals. So I'm sure you would have seen in the 1990s, the early 2000s, news reports about glow-in-the-dark mice that they've done by gene splicing jellyfish genes and mice genes together. My favourite was the Mighty Mouse that they created, which was a mouse that could run 6 kilometres, well surpassing the normal mice's limit of 200 metres. It was fitter, it was healthier, it was resistant to cancer, and it had all the good things going for it, the Mighty Mouse. In addition to altering the physical traits of animals with genetic engineering technologies, they've also altered the social characteristics. So there's a species of vole, which is normally polygamous. They would normally have lots of sexual partners. And they found that by altering a hormone receptor, it turned these voles monogamous. They would form these nice little lifetime bonds with other voles and happily raise children together. So this was all very exciting, but these techniques never had really serious potential to be used to alter the human genome. For one, they were very imprecise. They wouldn't just change the target gene, they would change lots of different other genes. And that meant a lot of the animals they were experimented on had serious side effects. They were also inefficient. They would often only change one of the genes that you were trying to target, and that meant that animals had to be inbred together to get the change effective. So they never had real potential as a clinically useful modifier of human DNA. But in the last few years, this has begun to change with the discovery of a new type of genetic engineering technology, which is now known as gene editing. So gene editing uses a system that is found in bacteria. It's part of the immune defense system that bacteria uses. The most common one that they use is called CRISPR-Cas9. That consists of two molecules. One is a scissor that will cut the enzyme, and the other is a sequence that will find particular sequences of DNA and latch onto them so the scissor can cut them. So in 2012, Jennifer Doudna and Emmanuel Charpentier published a paper in Science which showed that this system could alter bacterial DNA very efficiently and very precisely. This was followed a few months later with a group at MIT led by Frank Zhang, who showed that this same system could alter eukaryote cells, so the same cells that we have that have a nucleus. And these two groups are now locked into a bit of patent dispute over who's got the rights to the CRISPR-Cas9 gene editing system. And this dispute is going to be quite bitter because the potential of the system is just quite astounding. Although it's only been 5 years since it was first discovered, it's been used extensively in other animals to do remarkable things. They've made malaria-proof mosquitoes, drought-resistant wheat, hornless cows that can be used in agriculture. And they've also started to use it in somatic medicine. So that's medicine for our cells that we won't pass on to our children. Right now, there are various trials around the world using the CRISPR-Cas9 system to alter immune cells to make them better able to target cancer cells and target viruses like the HIV viruses. So it's done a lot of things, but relatively all of those uses in animals and in our somatic medicine have been relatively uncontroversial. What's been really controversial is the idea about using this same technology to alter our germline genes, our germline cells that we will then pass on to our descendants. In 2015, a lab in China became the first to use CRISPR on human embryos. Now, these embryos were unviable. They had 3 copies of every chromosome, so they were never going to be able to be born. But they still used them as a proof of sort of concept that you could use these on human embryos to reverse certain disease-causing mutations. These studies were hugely controversial. There was a big backlash. There was editorials published in high-ranking science magazines saying that this research should be stopped, that it was unethical. But despite this, research is still being conducted, and further studies with human embryos are planned or underway in the UK, Europe, and elsewhere. So I just want to talk about the reason why we might want to alter the human genome, and there's a few different types of reasons. The first is just in a type of research context. Only about 13% of human embryos survive to the first trimester of development, and we really still don't understand why this is. By using gene editing on human embryos, we can investigate the roles that particular genes are playing in early human development, and this can help us sort of unlock this mystery, and this may lead to just standard clinical treatments for miscarriage and to increase fertility rates. More controversially, we might want to use these in a clinical setting to treat genetic diseases. So we know there are simple Mendelian diseases like Tay-Sachs syndrome, like cystic fibrosis, that have devastating consequences and that are caused by 1 or maybe 2 faulty genes. It seems like it would be relatively straightforward to use these technologies to repair those mutations. And this could help couples who have genetic disease in their histories, but might not be able to use selection methods. More broadly, there are genetic components to lots of other diseases. So we know that sort of 30% of the world's deaths occur because of chronic diseases like heart disease, like cancer, like diabetes in those that are under 70. Now, we all have predispositions to those types of diseases. It seems possible that in the future, when we understand more about the genetics behind these diseases, we could use this technology to make us all more resistant to these chronic diseases and give us the best shot of living healthily into old age. We can even think about going beyond treating disease into increasing our other capacities. We might think a world would be better if people were smarter, if they were more empathetic, or if they were just happier. And it seems at least possible, as we understand genetics more and more, we'd be able to use these technologies to increase human flourishing. Now, those are some of the benefits. Obviously, there are lots of concerns with this technology too. Now, most directly, people are concerned about the research being conducted in human embryos. Embryos can't consent to this research. The results will be unknown. People think if a human embryo has a high moral status or is like a person, then this would be unacceptable research to conduct. More broadly about using them to fight disease and to enhance other characteristics, people are worried this will lead to a designer babies kind of a scenario where parents are picking and choosing the traits of their children like they pick and choose the accessories for their nursery. It seems like this might disfigure the relationship between parent and child when, before they're born, a parent can pick and choose the traits that their children might have. People think that this might balloon into other broader social problems. We might lead into a world where there are the genetics haves and have-nots, and that the rich have access to powerful genetic engineering technologies and use this to boost their advantages even more so than people in lower socioeconomic classes. And further, there are concerns that, well, we shouldn't be meddling with things we don't understand. By using these genes, we'll be affecting the germline, we'll be affecting our descendants for generations, and we might mess with human evolution. We might pick bad genes, we might lose adaptive potential in our future, and we could end up in a sort of genetic dystopia. So all of these issues sort of bear on the central question that we're going to be discussing today is, well, where should we draw the limits with germline gene editing? Should we ban all types of germline gene editing, not even use it for research purposes? Should we just use it for research purposes to understand human development, but draw the limit there and never use it in a clinical setting to improve people's genes? Should we allow it to improve people's genes, but only for therapeutic purposes, only to prevent genetic diseases and not go beyond there? Or should we draw no clear limits? Should we instead be guided about concepts like harm and benefit and try and use it to improve the world? Thanks.
Fantastic, great start. Alison, why don't you have first go of what your view of the benefits and risks and limits?
Right, I feel I'm a bit in a science fiction scenario here at the moment. And I want to bring it back down to earth slightly. I'm not a stem cell germline gene editing scientist. I'm a clinician who spent 25 years working in a fertility clinic. And I think it's important really to get some sort of very practical concept of what it is that we're doing here, particularly when it talks about human embryos. We see in Newcastle about 1,500 people who have a fertility problem each year, new patients. And although some of them will have problems, physical problems like blocked tubes or they've had a vasectomy done, most of them it's because there's a problem with these very early stages of human development when the time of fertilization and in the first few days after an embryo develops. And I mean, for obvious reasons, for obvious ethical reasons and practical reasons, it's really, really difficult to study that stage in human beings. And yet, that is the time when a lot of these problems that we've been talking about they might correct are actually occurring. So there is going to be huge benefit, potentially, for an awful lot of people who have difficulty conceiving if we can do the studies to try and find out what is going on at this stage. Just a couple of other points, because I know I want to hear what you're saying, not just what I'm saying. But if we're going to do studies on human embryos, then we're going to be using embryos that were created by patients who had IVF treatment and no longer need them for their fertility treatment. That in the UK is where most embryos will come from. It is possible to create embryos new, but obviously therefore the patients who are going to donate embryos for this sort of research, their views and their understandings of what an embryo is might be very different from yours as what I might call a third party standing outside looking at an embryo. We've done a lot of research in Newcastle talking to patients about their views about embryos. And if you want to discuss that, we're very happy to go through that with you. And then finally, on the same issue, if you work in an IVF clinic, embryologists are dealing with, hundreds of embryos, human embryos, every day, every week, I should say. And their understanding of what an embryo is, again, might be different from yours. And I'm very happy to talk to you about that again. It's quite important not to think about us as being mad scientists who want to do things in garrets in universities without any consideration for what the consequences might be. In Newcastle, we have a system whereby the research people doing research on the embryos are actually working in the department. They're not working on the clinical side, they are separate, but they work in a multi-user office, so they're talking to each other. So there is that context all the time between the two. And the attitude that they have to human embryos does respect their status very, very specifically. So just a few thoughts to throw at you on a slightly more practical level. That'd be, maybe we can have a discussion further on.
All right, thank you, Alison. Keeping the time very well. Andy, what about?
Well, I agree with Alison that we do need to keep this grounded in practical everyday realities. So my own research concerns trying to understand how genes regulate embryonic development. And we use CRISPR-Cas9 genome editing. But of course, this is partly, I guess most of you have turned up not to hear about the science so much as to hear about the ethics and to have a discussion about not so much whether these techniques will be used, but whether they ought to be used. If you ask scientists questions about new technologies and their applicability, you often get an answer which is a prediction. You will get an answer along the lines of, well, I think it will happen. But that isn't really the question we should be focused on now, is whether it ought to happen. I've had two experiences in the last 2 or 3 years that have really informed the way I think about these things. One is that I'm a member of the Human Fertilization and Embryology Authority, and along with Robin, I've been involved in assessing the safety and efficacy of mitochondrial donation techniques, otherwise known as 3-person IVF. And I think in ethics, you have to make comparisons and draw distinctions, or draw comparisons and make distinctions. And mitochondrial donation and the way that we went about trying to make it clinic ready in this country has a lot of lessons for us if we are ever to contemplate making changes to the nuclear genome. Remember, the mitochondria are not in the nucleus, and the DNA they have is separate to the nuclear genome, but it's very much part of the human genome. It's the human mitochondrial genome. So how we think about progressing with genome editing of nuclear genes very much depends on our experiences from mitochondrial donation. The other thing I want to say is that research is critical, that genome editing is being used in multiple organisms right now. One of the reasons why it's such a revolutionary technique is, unlike previous techniques, there are very few limits to the kinds of genome that you can edit. And that research will be critical if we're to make progress in understanding its potentials and power. But I do want to say again that ethics is really the thorny issue here. It's not so much the science, it's the what are we doing when we do ethics? And we need to think carefully about that. We need to think carefully about having those conversations right now before the science gets too far ahead of our ability to discuss it. So I think it's important that we discuss these issues right now before they're even technically feasible, so that we can have a discussion about the shape of that conversation. What is it going to look like? And what are the concepts which we need to employ? Because it will turn out to be equally as difficult as the science.
All right, fantastic. We're rocking on time. Robin?
I can spoil that easily. So first of all, before I start saying things, I have two sorts of conflicts of interest. So one is I'm on a study committee for the US National Academies of Sciences, and we are the final stages of writing a fairly big, comprehensive report on the whole topic. So what I'm going to say is I cannot say what the conclusions of that are going to be. So I'm just giving my own personal views at this meeting. And the other is I have a colleague in the same institute as me, Kathy Nayakan, who has a license to do human genome editing on early human embryos. And so she's a colleague of mine. So I agree with a lot of what's been said before. There's some issues to do with the science, and you can divide the science into the technical aspects And I agree with Andy very much that, you know, we need to think about, start thinking about what we can do in terms of using the techniques. There are some issues at the moment, so we can't do everything that we might suggest is possible now. There are some limitations. But I suspect that most of those limitations will get solved over the next few years. But there's also another aspect of the science, which is that if you're going to contemplate modifying the human genome, then you actually have to understand very well the specific gene sequences you want to modify itself, and in the context of the human genome, and to understand human genome variation, because we are all different, and modifying a gene in one individual may have different consequences in a different genetic background, different individual. So we don't actually, for many cases, know enough about human genes, genomes, and genetic variation to be able to predict what the outcome's going to be. So that's a word of caution from the scientific aspect. Also, I mean, Chris mentioned that there are somatic gene therapies are beginning now with these genome editing techniques, and they're really exciting, and they have a lot of promise to be able to treat people with a variety of genetic problems, including things like thalassemia, sickle cell disease, perhaps cystic fibrosis. There's all sorts of things that are being contemplated now, and there are some trials already beginning. People with HIV, AIDS, to mutate the receptor for the AIDS virus in their blood cells so that the virus can no longer attack their blood cells. And this is looking really, really good, beneficial to them. Now, you could propose, well, I want my children to be HIV resistant. Why don't I simply mutate the receptor gene in the early embryo, or whatever, how you're going to do it, so that my children be resistant? Well, we don't know what the consequences are going to be of doing that as a germline treatment. So that's a word of caution. However, if conventional treatments and somatic treatments for genetic diseases become successful, which we all hope they will be, then actually this may give an increased number of people who are surviving these often horrible, crippling genetic diseases now to reproductive ages, who will then want to have their own children. And so you then have, you're creating a problem in a way, that there's going to be a greater and greater demand for germline genome editing to actually deal with their problem, which is they don't want to transmit any mutant version of the gene to their children. So I'm quite happy to talk about aspects of the science and potential uses, and potential uses can be, as mentioned, correcting genetic defects. It can be introducing disease resistance or resistance to environmental agents. It can be making changes which exist naturally in the human populations, or making changes that are not typically found in humans, or not found in humans at all. And that's when you begin to get science fiction sort of discussions, and I'm happy to talk about those. Regulation is going to be, I think, something that's very important, and it will have to go before, or at least in parallel with, the ability to do things. So that's the science and the methods, and with the ethics. And so we need to discuss how you would regulate anything in this area. We are lucky in the UK, we have the Human Fertilization and Embryo Authority, which has experience in regulating similar areas of clinical science and clinical application, such as pre-implantation genetic diagnosis, which can be done on individual gene basis, or even for mitochondrial replacement, it's going to begin on an individual patient basis. So they have experience in that. That's not true. That situation is not true in many other countries, where the regulations are either a complete ban on almost everything to no laws at all. So how we deal with these issues when the science is global, the applications to be carried out anywhere is another potential issue we should talk about.
All right, thank you. So we have some roving mics, and now it's your opportunity to ask these eminent experts whatever you want. Do we have any questions? God, I've never seen no hands.
Hiya. I'm just wondering, talking about sort of our responsibility, whether we could or indeed should sort of pre-select for whether babies have diseases, like cystic fibrosis and stuff like that. Could you not argue we already do? A lot of Down syndrome pregnancies are terminated, and we do have the screening available for some such diseases, and often we do choose to end the pregnancy based on whether or not the child would have one of those diseases. Don't we do that already?
Yes, the answer to that, yes, is we do, and we select in various different ways. Some of us select our partner for good looks, or whatever, wealth maybe, or not. You can, as you say, you can have a termination if there's a problem with the pregnancy and it's not right. You can do pre-implantation genetic diagnosis, PGD, and select the embryos that you want to put back that are correct. So yes, that you could argue therefore that we are already in that position of selecting. I think selecting is different ethically from deliberately changing something. Selecting is just saying this is what nature's provided, we want to pick out the one to do, and you only really get into difficulties there where people decide they want to select for something that maybe the majority of the population might say is not I'm not trying to work for disability, ability.
Mm-hmm.
The question was raised about deafness. Would you allow a deaf couple to select for a child that is equally deaf? That starts to get into different sort of moral issues. But selection is different from deliberately manipulating something that's not artificial, but is different.
I mean, I agree, it's different, but the endpoint can be the same. And so,
yeah.
yes.
It's different because the risks are different.
Well, there is an important difference, though, that if you have two embryos and you have one normal embryo and one Down syndrome embryo, and you decide to select the normal child, you haven't benefited the child with Down syndrome. But if you take gene editing and you actually were able to edit out the problems with Down syndrome, you have actually benefited that individual, or have you? What's your...
Well, I mean, that's an interesting point for a number of reasons, because in fact, in some jurisdictions, you are not allowed to do research on embryos, or you're not allowed to manipulate an embryo unless it's a benefit to that embryo. And so, if you were correcting a genetic defect, then in those jurisdictions that actually would be allowed. And even not the current Pope, or I think it was the one before the last Pope, actually said that if you could correct a genetic defect in an embryo, that would be fine. He doesn't like anything else to do with research or treatments involving embryos, but that would have been fine.
In the process of doing IVF, you nearly always create more embryos than you're ever going to put back. Mm-hmm. And you are selecting on that basis anyway. You're going to select on morphological appearance, what it looks like, how it's developing, how it's growing, which is the one that's most likely to make a baby. So there is some selection process. It's not based on whether it's Downs or not, but you would probably know that the ones that are not developing it well, growing well in the laboratory, are probably the ones that are actually not going to make a baby. So there's some selection process there.
Andy, you wanted to come in on this?
I chaired a working group for the Nuffield Council last year looking at genome editing in a range of organisms. And there was one approach that we objected to, which is, I think, sometimes characterised as LC, which means you take a shiny new technology like genome editing and you ask, what are the ethical, legal and social implications of this technology? And that's one thing one approach that we didn't really like. We prefer to look at challenges that exist in our society. And obviously there are some big challenges. Okay, you only have to switch on the TV to see what they are. So for example, food security would be one, climate change, etc. And then you ask, in the context of those challenges, what are the range of possible solutions, or indeed at least a range of possible approaches to alleviating that problem. And that way you don't just focus on the technology, you focus on the technology in a range of contexts, which I think is important. So for instance, in the case of disability, it's partly about focusing your mind on what does this approach to eliminating the birth of certain children say about the people who live with those disabilities. And this is a common area of discussion that many people, it's called the expressivist concern, that many people are concerned that those technologies express an attitude towards individuals living with disability that we don't want, a parent might not want to have a child with that disability. Now, personally, I don't see that there's a logical link there. It seems to me that we can show love and compassion and concern for every living human being, including those with disabilities, and yet at the same time offer a parent the right to make a choice about whether they want a child with that disability. I don't see a logical disconnect. Many do. And this is the problem with ethics. We have to reconcile disparate perspectives somehow. But the only way of doing that, I think, is to respect each other's perspective.
Yeah. I mean, in fact, specifically on that point, so the sort of disability rights has gained momentum is exactly the same time as society has generally made provisions for people who are disabled in some way. From everything from better access to buildings or transport to things like the Paralympics. So they are disconnected.
We should take some other questions. Hands just here in the front, the gentleman, and then the lady behind.
I have two questions. The first one has to do with scientific or clinical responsibility. So this is an argument that has been raised recently more and more, which is particular to the two aspects that Christopher has mentioned, which is correcting existing disorders, genetic disorders in the germline with the aim to eradicate potentially those from the population, or preventing diseases, or introducing certain changes in the genome that would help us sort of with acquiring certain disorders that we actually learn very much about them, such as cancer or cardiovascular disorders. And the argument that has been made recently more and more is that once all the regulation and the safety is in place for these technologies, will it actually become unethical not to make these corrections? So this is the first question. And the second question is an interesting point that was raised at the Keystone Precision Genome Engineering Conference that I just attended. The question when it came to germline editing of the humans was, what makes us special? Why do we actually ask this question about humans when we obviously have no problem in creating all these modifications, and this is going to happen more and more in the future in all kinds of animal species as well as plant species. So I was wondering what this panel thinks about it.
Chris.
I think there were several important points there, but one was the last point, which I think made everyone's ears prick up, was what's so special about humans? If we're busy altering the genomes of numerous organisms on the planet, what's so special about humans? I mean, I take it that there's an answer to that which sounds flippant, but it isn't really. I mean, what's special about humans is I would only ever consider marrying one. Most of my best friends are human. I am one. It sounds flippant, but I think it's not. I mean, I think that being a human is not just a biological category. Okay, being human is special in the sense that I've only ever had meaningful relationships really with humans. I love animals, but as I say, I wouldn't consider having one as a best friend. So I think there is something special in being human, and I don't know how to say anything more profound than that, other than I wouldn't marry a dog.
So I think the first question though was, if this was effective, wouldn't it be obligatory? Which I think is a very interesting question. If you're able to cure cystic fibrosis, would we actually allow people to refuse? I mean, we wouldn't allow a parent to refuse a blood transfusion for their child bleeding to death. In fact, courts regularly overrule Jehovah's Witnesses who refuse for their children. If a parent refused to have their embryo gene edited and there was a treatment for cystic fibrosis, would we allow that?
I would just say that there's a principle in medical practice that the decision of whether to treat or not is taken by the patient with informed consent, and I can't see anything in this field that would override that in terms of saying society says you have to.
Really? I mean, you can't refuse life-saving treatments for your child. The consent is the courts will authorise so if you have a life-saving gene edit, the extension would be the courts will override parents' refusal. The courts will give consent.
I think there is a difference. This has never been challenged legally, but I suspect when you're talking about a potential child, not an actual child, you're in a different legal scenario entirely, and I think that makes it very different.
But we should distinguish too the legal question about whether it's legal different or is it ethically different, I think is the important question. Is it, if you've got the opportunity to save it, to correct a disease-causing mutation, and if you fail to do it, that child will have a terrible disease. I mean, what's the ethical difference between doing that and just not treating them once they're born?
I think one difference would be the social consequences. So, for instance, if we're serious about the idea that any potential parent should prevent the birth of a child with a disease, the more that we sequence genomes and the more that we analyse genomes, the more we realise that we all carry hundreds of deleterious gene abnormalities. And in combination with our partner, you might end up being homozygous, sorry to get technical, but 2 copies of these defective genes for potentially hundreds of different genes. And so the consequence of that would be actually that we would have to have systematic, whole-scale, societal, technological intervention into reproduction. So it would be almost inconceivable that in the future we could allow anyone to just have sex and have a baby. Now, I suspect that perhaps some of you wouldn't look forward to that future. And that's, again, slightly flippant, but I think we need to think our way carefully through. The more we learn about genomes, the more we realise that human reproduction is already something of a disaster because of the ways in which human biology has evolved. And to try and intervene technologically will require, as I say, wholesale social re-engineering of what it is to be a human, coming back to your question.
But I mean, I agree with you in many respects, because I mean, if a child was born with suffering from a genetic disease that could have been avoided using the technology, then would you not then the parents could be sued for the...
I think the other thing to remember though is that the patients who have these problems, and we come to this issue with the mitochondrial side of it, the parents who have a disease in their family, they know the consequence of that disease for their children and for their family and the wider family much better than anybody else does. And in reality, they are the ones that are asking for treatment. It's not the other way around where society might force it on them. I think the idea that they would deliberately refuse treatment is less likely.
I have a line of 4 questions now. I've got the lady here, then you, and then you. No, just with the white. Yep.
Hi, this is a question that's probably more to do with the use of gene editing in preventing multifactorial diseases, such as non-communicable diseases. How would you respond to the statement that gene editing and thus affecting the course of biological evolution is less of an act of God nowadays, seeing that cultural evolution has pretty much superseded biological evolution in reasons behind people contracting or developing such conditions? Do you think that diminishes the importance of gene editing and the role of genetics and the role of biological evolution?
Yeah, I think that's a great question. I like to think about it in sort of 2 ways. And the first is, well, what is the difference between the massive changes that we've seen through culture to massive changes that we might see through genes? We know that certain technological developments have really changed what it means to be human, such as reading and things. It changes our brains massively, increases our cognitive capacities. Now, people don't sort of think about those changes as anything icky or sort of wrong with them. People think that's sort of progress, and you might think, well, what's the difference about using genetic manipulations to also increase sort of changes? But I take it your point was, well, we've already got these great cultural, we can treat diseases culturally now, we can do vaccines, we can isolate people. We don't necessarily need genetic resistances to diseases, and therefore maybe gene editing will be less essential. But I think there's also the flip side of that is that we might have cultural sort of led sort of challenges that maybe gene editing will be the best response to them. So for instance, if you take the changes, the challenges of climate change seriously, we might need to think about really radically altering our nature to be able to handle some of the changes that might come. And that's not just our physical nature, but also maybe our sort of moral nature, our way to be able to cooperate and to think long-term with other people.
Follow on by saying that with the idea that, particularly when you talk about multifactorial genes, is that that determines absolutely your fate in life, is missing the point that actually your lifestyle is probably going to have a much greater impact on your long-term health. If you eat too much and you smoke and you drink too much and you're overweight, that's going to have a hugely greater impact on your life than one of the many multifactorial genes that we might be interested in.
So, two slightly unrelated comments to that. So one is, you could take as an example the ApoE4 allele of ApoE genes. So that's ApoE4 allele is associated with a higher risk of Alzheimer's disease. So you could say, well, why don't we edit that allele to now become ApoE2 or 3 with a much lower risk? Well, first of all, we don't know why that's possible theoretically. But we don't know why the ApoE4 allele is present at such a high frequency in the human population. So it could be because it confers some advantage in another respect. So you may have a high risk of getting Alzheimer's, but you're cleverer or something, or you're resistant to something else. Or it could be a gene that actually is on its way out of humans because non-human primates, like chimpanzees, only have an ApoE4 version of the gene. So it could be something that humans are in the process of losing, in which case perhaps it doesn't matter and it would actually be speed up, if you like, evolution to a better state of humanness if we were to change it. I mean, you're getting on into the area a little bit of enhancement, of course, as well, because I don't know whether you intended to go that direction. You didn't. Okay, well, let's leave that till later.
Okay. Oh, sorry. I thought we've got a line, so we should keep rolling. The lady at the front was next. We've got a line of about 5 questions at the moment. So.
Thank you. Fascinating. I've got a lot of questions, but one I'd like to ask, perhaps because we can't ask all the questions. I mean, I don't like animal experiments to begin with, so I don't like the idea that all this was done on animals. Was any ethical discussion ever made about that? And secondly, I heard, unfortunately, the end of a programme, which I want to listen to the whole of on the radio, and what they were about this, and what they were discussing was the time period. At the moment, you can't do anything on embryos only up to 14 days, and there's a suggestion that this should be now prolonged up to a month, and the argument was that an embryo of 14 days really is not kind of, anything sort of, a lot of them die anyway before that stage, so why should we worry? Well, I think I do worry, the thought of a month-old embryo being, messed about with. And I'm very interested in all this discussion about evolution, you mentioned it, for a better state of humans. I got into this, interest in this, because my poor grandmother was involved in eugenics in her day because she wanted a better state of humans. And this was, of course, completely, considered to be totally unethical and, something that one should never even think about. I can't help feeling she'd be awfully glad to think that you could now manipulate a gene and prevent the congenital diseases that she was worried about from being perpetuated from generation to generation. That's another story. But perhaps you could answer about the time period, because a month-old embryo, I mean, that's quite, you know, anyway.
Okay.
And the other thing, sorry, one more thing about genetic resistance. Since we've got, you know, problem with antibiotics, maybe we might need to have this covered.
Go on. Well, you go. 28 days, 14 days. The whole processes of human development start at the time of, well, just before fertilization in terms of the egg. And lots of different things are happening at different stages. And there's a lot we need to know. I think some things happen day 2 and some day 8 and some day 14. There are some things that happen beyond that which we don't know about. And I think probably if I had a crystal ball, I would say that at some point in the future we need to look at those later stages. My personal view at the moment is that there are still so many things we need to look at the earlier stages that we need to be getting on with doing that before we move to the 28 days, but we need to have part of that discussion now. Can I also just quickly go back to your question about humans and animals? Not that I want to talk too much about the animal work that preceded this, but just to explain that human conception, the genetics of human development is so different from most animal species that actually we have to do this work in humans. You are arguing against doing it in humans, but there are some that would say that we ought to do it in animals, doing animals, we ought to do it in animals first. But
precisely for that reason, I don't like animal experiments, because humans are so different from animals.
When we were developing the work for the mitochondrial transfer work, we were initially doing work in animals. But it became clear that the work that had been done in America with primates, even that was so different than the human side that we had to say, no, we can only do this research on the humans. We need to go straight to that.
So Chris, is this eugenics? The question, or there was another question about the return to eugenics. Is this the return to the eugenics of the Nazis and the 20th century?
Yeah, well, it really depends what you mean by eugenics. It's certainly not a return to the eugenics of the sort of Nazi period, which was based on the concept that there's one ideal sort of genome that we should all strive towards, and that we can use coercion to do this, and that the idea is hereditary, that there's one gene for intelligence, and you've either got it or you don't have it.
I would say that was not my grandmother's interest in that.
Yeah.
She was concerned about the people who were unfortunately having, children with diseases which could have been prevented if they hadn't given birth, really.
Yeah, so the other side to say is that, well, we might just mean by eugenics an attempt to improve hereditary, or attempt to improve innate, and in this case it's eugenics, but eugenics is good, and we do it in other ways too, just through normal medical practice.
So I think it's a big difference between state-directed programs, and as Alison was saying, that if it's up to the patient to decide, then that's not really, I think it's not the same as eugenic programs as we worry about.
But you just said before, in response to this gentleman, that we should do it if it's to do something like cystic fibrosis. So can't you see that this could quite easily slip into being something that isn't so free?
Should be offered.
It should be offered.
Should be offered.
But it should be up to.
I think there was a gentleman over here. I've got you on the list now. Yeah, just back there with the glasses.
Thank you. I'd like to ask you guys a question about the complexity of the genome. We're talking about single mutation in single genes here, but most genes don't have one effect, they have several. And if you change one thing, cystic fibrosis, for example, you could say, well, that's very bad, but it actually also protects against measles, so maybe it's very good. So I guess this was the point you were making before. Thinking about how we would do this, let's just say it's possible, how would you take the complexity of the genome and then say, oh, this is the best genome, this is what we should do to get more intelligent children? How would you do that? Because I don't think it's possible.
We will, I mean, there are all these big genomics projects, the 100,000 genomes or whatever the latest number is, where you're linking human genomes with health and lifestyle records, and so we will learn an awful lot more over the years about what the particular combination of alleles at particular loci how they contribute to our health and our happiness or whatever. And so I think ultimately we will be able to do more and know what the risks might be. But I think at the moment, well, you might not have to change thousands of genes. It may be just a few, but at the moment we don't necessarily know, because we don't necessarily know what all the genes do in sufficient detail. I mean, there are some genetic changes you can predict precisely. If you're carrying, say, the mutation that gives you beta thalassemia, that's bad. It can protect you against malaria, but if you're not going to be, subject to likelihood of infection by malaria, then why would it matter if you correct that defect? So, I don't see a problem personally with that. But there are other things, like I mentioned, ApoE4. We just don't know. So I wouldn't advocate at the moment changing that version of that gene. But in the future, we probably will know what the consequences would be because of just looking at natural variation. You're looking very doubtful.
Well, one gene has a very, very, very small effect when you look at these GWAS studies.
Yeah.
Like the gene has a very, very small effect, and it might have several functions. So if you take the protein of a gene, on average, a gene codes for interacts with 700 other proteins. So how are you going to predict, if you need to change thousands of genes, how are you going to predict what the effects are? And then basically what you're saying is, well, we'll just have to try it out then, or would that be unethical?
If you're making a change that is going to be mimicking something that goes naturally in the human population, then are you doing anything particularly dangerous?
I think it's absolutely right, though, to raise the problem of complexity. One of the reasons why I don't like the word gene editing is that it focuses on genes. I prefer genome editing. And that's because it's difficult to know what genes are. I mean, they're actually quite slippery little things. To know where they start and where they end isn't actually all that clear. And whereas genomes are a little bit easier to get a grasp on, as long as you've got a grasp on what a species is, although I acknowledge that even species is a bit slippery. And I agree that what we've learned since Mendel, is that there's no such thing as a gene for X. We also know that mutations in genes can sometimes have pathological consequences, but they're dependent on the genetic context. So it's true that in one genome, a mutation will result in a disease, and in another genome, that mutation seems to have no harmful effects at all. And so what this is telling us, of course, is that genes cooperate, that genes interact, and they function together. And so what you're doing when you introduce a mutation is to disrupt a network. And it is often very difficult to understand how the mutation will reverberate through that network in some kind of deterministic, predictable way. So you're absolutely right to raise that. But of course, I would want to make the plea for our ignorance, because I'm a geneticist, and I need money to do research. And there's no way, as Robin points out, that we're ever going to make progress in understanding these phenomena unless we study them.
But can I just ask a quick question to follow up, because people are often sceptical about the capacity to make large-scale changes to the organism in a safe and effective way. But Chris raised this case in non-human animals where you've created fluorescent monkeys or fluorescent mice or fluorescent rabbits. And by introducing genes that aren't normally in the human genome, you can make profound changes. So Annie mentioned food security. Humans cannot metabolize sunlight. Plants can. You could introduce genes for chlorophyll from plants into humans, create green humans that metabolize sunlight. You could create enormous changes. Now, Robin, do you think that is actually possible in humans in a way that could be done using gene editing?
It's not possible now, but it could be. And you could say, well, why not? And, or to at least allow people to eat gluten, for example, would be a useful thing. Allow people to, we may, apart from climate change, or actually one of the consequences of climate change, maybe we need to leave this planet. And so long-distance space travel, you're probably going to have to change people's genetics to allow them to survive long-distance space travel. So
Okay, I think, Marcella, you were next.
That would sound to be state-directed, though.
all the volunteers.
Thank you. I have got a question concerning the distinction between selected and changing. I see there is a distinction here, but usually you select one embryo out of 5 or 6. But now there is some speculation about the use of induced pluripotent cells to create many eggs at a time. It's been done last year with mice. So in principle, you could have hundreds of embryos, brothers and sisters. And as the technology for sequencing gets cheaper and cheaper and quicker, so you could in principle have hundreds of embryos and select one of them, and that would pretty much amount to changing. In the end, the result would be not very different. In that too would be a kind of eugenics. And how do you see that?
Okay, there's several issues in there. I think the first thing to say is that creating new gametes, new sperm, new eggs from iPS cells is not impossible, but I think it's still somewhere down the line. It would be illegal in the UK at the moment, so we're not talking about something that's going to happen instantly. You could be in a situation even now where you could maybe take a woman's ovary instead of developing her eggs in the normal way for IVF with medication for 2 weeks, and if you take a bit of ovary, you've got huge numbers of eggs from her, you could potentially create lots of embryos. So the issues that you're raising from that perspective, actually having a lot to choose from, are actually not new. From IVF from fertility point of view, we need to create several eggs, have several eggs to create several embryos to have a realistic chance of success. Even then, the best success rate is going to be maybe 25% per cycle, 65% over 3 cycles. So we do need to start doing that basic research now, which is where it's coming back to drawing you back from these questions about what we could do in the future with gene editing. We need to be doing research now, and we need to do that research now in these early stages of development so we can understand what's going wrong. We can understand why so many of the embryos that are created normally and in the laboratory from humans are genetically abnormal and have no potential to make a baby. So that research, I think, is really important, even though it might be a slippery slope.
Okay, but I think you were next. Yeah.
Thanks. This is a question for the last night in the world before Trump. Whether the first or last Trump, we have yet to find out. There have been 2 initially benign technologies since the war developed. Information technology has underpinned our communication, but in the 1970s or so, grumpy Russian and Bulgarian cyberneticists invented the bug, and out of that, eventually, we have the machinery of cybernetic warfare. And I wonder whether there's the same potential progress, which was slightly emphasized in the last of the presentations. There's the same potential process in the development of gene technology, that eventually, in a very unregulated world, there will be genetic warfare in the form that we've seen cyber warfare.
I hope not. I think that's why we are, in a way, why we need to have these discussions, and we need to have the public discussions and public understanding and of what's possible, and influencing governments to have proper regulation. And so the regulation in the area of assisted conception is incredibly variable throughout the world. So here in the UK, it's good, robust, and I think it would be very hard to imagine that here, at least unless there were big changes, that you could propose to do some form of enhancement or modification of future generations to make us resistant to a disease, a virus or something, which you could then kill off, French people or whatever with. Just I have many good friends who are French. So I was just thinking of our closest neighbours. I mean, you could take it. I mean, you could let's say right, North Korea. Let's take. So that's an environment, you know, a particular regime where you can imagine they would be having these thoughts. I think it would be very hard to do it in a way, and certainly it would be it would take a long while to modify people in that way. And you would know about it, and it would lead to an arms race, probably in a very weird way. And I don't think it's practical. However, it needs to be debated, and I think if the public are aware of what's possible and what's not possible, and they can influence their governments to have appropriate regulations, then that would be very helpful. So in the US, the regulations, we're talking about Trump, the regulations for assisted conception and everything are a mess. So actually you can clone people in some states in the US. Right? So it's already a problem in some serious countries, but
I think from that point, you once you've got an understanding in science, you can't go back.
Yeah.
The knowledge is there, and what we have to do is discuss how we manage it and how we progress from there. I think I'm less concerned in this area because most people want to reproduce in the normal way. It's much more fun. And the idea that, I mean, if you're talking about cyber viruses and things, you know, they can go around the world in an instant, just about. Just try doing gene editing, doing all those processes. It's very complicated. It's never going to happen any large scale. It's going to be something that was only going to be possible to be done with people who've got specific skills in very small numbers. So, I mean, I would agree. We've had information that just this week that the mitochondrial transfer techniques have been carried out in Ukraine on people who've got an infertility problem, even though there is absolutely no reason for doing it for infertility. It's not allowed in the UK because there's no evidence that it is beneficial and safe relatively. But Ukraine doesn't have any regulations. Mexico had no regulations. So there is the likely possibility that some of these things might be, will be, I would say, carried out in countries that don't have regulations. And I don't think there's much we can do about that. But actually the worst thing that could happen would be that it would be stopped as a scientific progress, because that would just drive it underground.
I'm worried, I think, more about, in terms of unregulated uses of the technique. You have, so in the stem cell field, there are various clinics that are set up to offer stem cell treatments which are completely bogus. And the clinics who offer these treatments make large sums of money from desperate patients or their families. And you can imagine the same thing being offered in the genome editing world. That, you know, I could say, I'm going to set up a clinic, I guarantee that your children will not get Alzheimer's disease. I make a fortune. And, you know, by the time you know whether your children are getting Alzheimer's disease, I'm long gone. So it really scares me, that aspect, which is why we need to have, I'm afraid we need to have regulations in this area.
So I'm kind of missing the answer to the gentleman's question, because before you told us that we were able to gene edit somatic cells to provide resistance to disease, HIV you mentioned, but you could imagine that the North Koreans or the Chinese or whoever your favourite bogey man is, are engineering resistance using somatic gene editing to Ebola. And now they have a perfect biological weapon that their population will be resistant to. And so what you say is, no, we need more regulation in the UK to protect against this. No, I'm not seeing how our regulations here will protect us against North Korea.
We need other countries to go.
Well, that's just not going to happen. So surely we should be pushing on with the research and having less regulation here, so that when that does happen, we actually have the means with which to respond, rather than being caught back-footed by regulation.
That sounds like an arms race to oblivion to me. Yeah. Well, it seems to me that this is an example where discussing the technology is something of a red herring. If we live in a world where our governments think that the way to deal with global problems is to annihilate other nations, and that's the problem. It's not genome editing. No. It's not science. That's the problem.
Yeah.
So did you have your hand up before? The gentleman just, did you have your hand up earlier?
Yeah, I guess it's a question a bit in line of what we were just discussing. My question would have been, how feasible is it that a terrorist organization might use some of these techniques to weaponize, for instance, bacteria? But also then, to what extent controls on science, on regular science, might slow this down or even prevent it. I think there have been discussions about moving some of the scientific discussion into a more closed-off area instead of making it all open access.
I think keeping things open is the only solution. If you do things behind closed doors, it gets more of a problem. Of course, you can use any of the technologies, this sort of technology or others to make bioweapons, and that's a concern. You can, the technology in a way is so cheap and simple. It's turned into a garage science. So there are people using CRISPR genome editing in their garages.
Or their garages.
Yeah, depends how you pronounce it. And they're doing this, of course, on bacteria or yeast and things like that. To do it on human embryos or gametes would be very challenging in your garage.
Well, but I think as well, if you're talking about things like altering bacteria, you're actually potentially trying to create a weapon of mass destruction. Well, we actually already have weapons of mass destruction in the world, and we manage them in different ways. So I'm...
Well, it's true, but an atom bomb is very hard to come by for a terrorist organization. Technologically, it's extremely difficult. And I was wondering if these particular techniques are technologically, at least, let's say, in a time frame of a decade or so, much easier for them to apply than building a nuclear bomb.
Yeah, you've got to protect your own people from whatever bacteria you might want to manipulate. I mean, it's betting out of my field slightly, but...
Yeah, I mean, you can use anthrax. I mean, you don't need to manipulate that at all. But it's an area of concern, and I can tell you that there are people looking into this as an area of concern. But I can't tell you what they're thinking of how to control it.
Yeah, we contacted the Ministry of Defence to ask them to comment on it, and they didn't.
Some people think HIV came out of the laboratory.
Okay. Yeah, let's, yep.
I don't believe that.
Yep.
So I'm a bit skeptical about what you're saying, that you say you don't think like North Korea or something, they would be able to do like large bulk changes in human to like make soldiers or to gain some kind of strength. But like we're discussing a technique that had been developed 5 years ago, and like 6 years ago, you would probably sit here in a panel discussion saying, I don't think this kind of gene editing or genome editing will be possible. So it's a bit hard to say that you say, I find it hard to believe that you say, I don't think it's possible that they will do these large changes, because we are in a world where changes or like new techniques have been developed so quickly. So it's hard to sit down and say, yeah, I don't think they can use that, because maybe there will be a new technology and then it will be accessible.
There are ways of doing. We can think of ways of doing it now, which either can be done more rapidly or less rapidly. So there's a method called gene drive, which could be used to introduce genetic changes through generations. So it would take many generations for whatever genetic change to spread through, be present in sufficient numbers of your population. But that's possible. That's what's being proposed, for example, to deal with malaria, is to use these gene drive methods in insects. You could also, in theory, use viruses to make genetic changes using the genome editing methods with the viruses. You would have to develop a virus that's going to infect germ cells, but that's doable. So yeah, no, it is doable. It's just I don't know. I don't know. I'm not saying it can't happen.
It's what we would have to do to prevent it from happening. I mean, science operates by the free movement and exchange of ideas. It simply, all of us publish in open access journals, which you can get access to for nothing. That's just the way it works. The idea that we could overhaul that global practice of science to have a much more secretive process globally, and obviously we know that secretive science takes place, but the idea that it could become one massive secret global exercise such that there wasn't the free flow of information, it would completely stifle scientific innovation. It would be ironic that we'd be losing the kind of freedoms that we're meant to be fighting for, I think.
So you need to keep the time frame in context here. If you want to create a mass population with a specific gene, I mean, for a start, if you're going to get an army of soldiers, you're talking 20 years from birth, and you've got to have enough. The logistics of trying to do that, to create a super army, are not realistic.
Okay, we've got quite a long list of people. I'm trying to keep up with whose hands are coming up, but I'm taking in the order which they appeared. So I've got you two on the list, but you've been waiting a long time.
There's got to be one subversive question this evening. I get the feeling that all the panel are both appalled and excited in equal measure by their work. I wonder if any of them feel they would benefit from taking time out to paint or write poetry or philosophy.
I think yes, and but the Medical Research Council don't offer sabbaticals to do that. In all seriousness, I do think that if we're talking about ethics, or we're talking about how do we cultivate an ethical disposition, then I think that novels are very important. Novels are an overlooked resource in cultivating new and interesting ethical perspectives. So not everything, I think, is necessarily achieved by rational argumentation. A brilliant argument can leave someone cold. A passage of prose might move them to change the way they behave. So that's my sensible answer to your question.
I mean, there's a lot of science fiction novels painting very dystopian futures, which a lot of the concerns that people have are actually probably come from the... Yeah, exactly. That's where a lot of the views, the concerns come from. I do paint in my spare time, not to have any.
I think you were
I
next
do a bit of gardening too, but can I just add the question about part of what you're suggesting is related to timescale. When we were doing the mitochondrial transfer, getting permission to do that, we've only just got down to the stage of position. We first talked about that 16 years ago. So during that time, it has been a constant discussion, debate, political argument, stop, rethink as it's going along. So it's not as if something, oh, well, here's something, right, we'll do it tomorrow. Certainly that took a long time of reflection and consideration before it actually happened. Some would argue far too long, but that's a different issue.
Okay, so you're next in the front.
Right, so you've mentioned a couple of times that in the debate between state programs and being a patient's sort of autonomous choice. Sorry. I thought about the sort of clinical research setting and the Declaration of Helsinki, where consent is obviously very important, and external pressures that mingle with this process of consent should be prevented. And when pressures such as, for example, a financial reward are too high, then obviously research proposals are rejected. Now, in sort of the context of a society where the pressure becomes so incredibly high, to have your children, screened and edited to, because certain characteristics you just don't want for your children, is it an actual choice? Is there autonomy involved there? Is it a pseudo choice?
My impression from many years of clinical practice and obstetrics and gynecology is that people actually don't want to select things for their baby. They just want their baby and take a chance at it. And there's nothing in the patients that I've come across who said, I have to have a child with green hair. Okay.
Have you seen Gattaca?
But that sounds fiction. I'm talking about what people now who are reproducing now are actually saying. They are more interested in saying and taking a chance on it, because that's the way we are brought up to do it. Now the question then rises, what if you give them a choice? And I suppose the easiest section is sex selection. You give people a choice, do you want a boy, do you want a girl? And that becomes a difficult one, because I don't in principle have problems with that sort of choice, except that in certain cultures and for certain people, it's sex discrimination. So, you know, there's a balance to have then. But I think it'd be highly, highly unlikely that social pressure would actually change that. Most people, when they conceive, don't even think about the fact they're going to get pregnant. It happens behind the bicycle shed, okay? You know, it's not something that is a very positive decision for most people.
Chinese women are being drowned though in China because, and so there is sort of a perspective of societal pressure. I was referring to Chinese children, you know, with the one-child policy. Obviously, there is some, you know, you could imagine that even though Gattaca is science fiction, that in a situation where this would be possible, would then, you know, the pressure becomes so high because it is possible that these sort of situations arise, and would that then be a reason to how would you?
But the situation in China is pretty unique, and you have a situation, say, in Beijing, where there are hardly any neonatal units, because if a baby is born and is not well, you let it die. Because if you're only going to have one baby, you want to have a baby that's a healthy baby. So you're talking about actually a society that has a completely different view about human life and what it means. And I think we have to be very careful that we don't take the ethical views that we have that are sacrosanct in this country and assume that in another culture they are equally important. We've got to be a bit careful about that.
He is right that there is a massive market in the US for desirable gametes. You see people who are infertile, desperate to have a child. In the US, when you have money and you can do it, people buy an egg from a beautiful, blonde-haired, athletic, Ivy League woman for $100,000 US.
Yes,
so they actually will buy better gametes.
No, those are people who need to have gametes to reproduce.
Yeah, but if they've got the money, then they're doing exactly what you say. They're choosing to have what they judge to be better children.
Okay, so we have a patient that comes along that he's got no sperm, you need a sperm donation. They're given a list of who the donors are and they're given some basic characteristics, they're making some choice. Okay, if you're in the States, you can go to your Nobel Prize list and you can get a choice. It's a different sort of choice. But that's only a tiny, tiny number of people in a whole population. We mustn't assume that is something that then is going to be translated to the whole population.
All right, we better keep moving. I think you were next. Okay, then I think there was up at the back, the lady with the glasses.
As a scientist, I would really jump and say we should go ahead and do it. We need to make the mistakes and learn, because this is the only way science would advance. But if I want to smooth it a little bit, I do think we need to have somewhat of loose regulations, because some of the human nature is if we put too many regulations on things, people would start doing it illegally. So if you think about abortions, if it's legal, then people would do it in a regulated way. We make sure that no mistakes happen or fewer mistakes happens. And the same, I would assume, would be with genome editing. If we are putting so many regulations on it, and then it goes to the black market, so we lose any control.
I agree with you. I think what you need is appropriate regulations, where decisions can be made based on the obvious things, whether risk versus benefit. And they don't need to go to the black market in the UK. They can go to another country and do this thing. So that's the thing that worries me more. I think it's having appropriate regulations, not over-regulation. Here, we may disagree a little bit about the value of the HFEA, but I think they tend, on the whole, to do a reasonably good job.
You're looking at a panel here of the regulator and the regulated sitting next to each other, and I think that probably tells you a little bit about how it works in the UK. We have an Act of Parliament that is a very good Act of Parliament, because it doesn't try to say what is the moral status of an embryo. It doesn't think about the embryo in terms of a physical, scientific definition or an ethical decision. It says this is an embryo because that's what we want to call it in law, and it defines it that way, and it defines very clearly what we can and can't do. And it also says very clearly what the penalties are if we don't abide by it. And we can go to prison, basically. It's a criminal offence to not comply with the rules and regulations that are set there. We can disagree a bit about the authority, maybe, but the act I'm very happy with, because it's allowed a lot of progress to be made in a way in the UK, which is not, we're not at loggerheads with each other. We go about it. I know what the rules and regulations are. I know what I can do, what I can't do. The fact that I would have a license to do it protects me from those people who might want to challenge me in courts, because I can say I've got a license. So it's clear. Then you have the situation that you referred to, say, in the States, where it becomes a free-for-all. And in fact, the ban on state funding for it actually drove it underground. It drove it into the commercial sector where information is not published. We don't know what is happening. So I'm not opposed to regulation in a very consensual way.
I think in a liberal, I think we're in a liberal democracy. Might switch on the TV tomorrow and find we're not, but I think we're in a liberal democracy. And I think that, democracy is pretty much all we've got if we're going to navigate through, ethical, moral pluralism. We undoubtedly live in a pluralistic society. Now, obviously I'm right, but the way forward is for me to listen to all these disparate viewpoints, and regulation is one way of negotiating that, and also public debate is one way of negotiating diversity, and we don't have many other options other than to acknowledge diversity, but also try to deal with it some way and cope with it some way. And regulation is one way that we can cope.
Okay, the lady with the glasses has been waiting a while. Okay, and I've got you. Any other? Yeah, sorry.
Sorry.
Yeah, that's all.
Hi, I wanted to ask if you could expand a bit more about the socio-economic aspects of germline editing, in particular in comparison, not just to if we ban it here, people will go to another country, which I think we've sort of touched on, but also compared to somatic modification, because you can imagine that with somatic modification, a patient has it, but then all his descendants will need it, which could potentially create a much bigger economic gap between the families that can afford it and the families that cannot.
It's definitely a concern, and it's one of those things that if you sit in a room with scientists and social scientists and discuss any novel biotechnology, there are various, it's almost choreographed. You could predict the moves that everyone will make. And the social scientists will immediately remind you, certainly the sociologists will remind you of things like distributive justice and social inequality and entrenching all of these isms which are so objectionable. And the real prospect that we need to ensure that does not happen. I mean, of course, there is this term genetic exceptionalism, which I think Chris kind of alluded to, which is the idea that there are other ways of influencing future generations and our society that are not genetic. So, for instance, yes, I think it's right to be concerned about social justice. I mean, I'm concerned about social justice. I don't like inequality. I don't like the fact that half the world's starving, where the other half of the world's so fat it's suffering from diabetes. That's crazy. So how could you not be concerned about that? Knowing what to do about it, of course, is another question entirely. We live in a society where it's acceptable for people to spend thousands of pounds on their children's education to buy them an advantage. And an education will almost certainly change their biology, because it will probably rewire their central nervous system. So we're already impacting on biology using money in a way which is socially unjust. So this technology needs to be seen in that context, I think.
Okay, I think we've got to wrap up. So the last question, the lady there has been waiting. Sorry, mate, just there. And just a very brief answer, because we...
Just to take it a bit back to the science, if we do go ahead and do this gene editing, do you think we could be shooting ourselves in the foot a bit? Because if we're reducing our genetic variability within the population, does that make us more susceptible to diseases in the future? Like, I don't know what you think.
Well, Danielle's my PhD student, so I'm not answering that question.
I think, well, I mean, I think you would have to alter the genes of a lot of people to get to that state. But and then we are of course, there's always new mutations that are occurring. So I think it's unlikely we're going to take a lot of effort, I think, to reduce genetic variability in humans sufficiently. So personally, I don't think that's a big concern.
Yeah, I suspect a mathematician could work it out for you, but I suspect the odds would be very, very long.
Okay.
Yeah.
All right. So I want to ask the panel to answer this question, which is the question that we were posed at the beginning. Gene editing, should there be limits? 1 minute, each of you. What, if any, should the limits to gene editing be? Robin. Literally 1 minute.
I think a simple limit would be you could be restricted to make changes which are already present in human populations, so that you don't go outside that. But that's qualified, I think, in terms of only that's the normal situation. So if climate change, space travel, whatever, it becomes necessary to change it, then I think everyone would be happy to accept changes even outside those limits. So that's
good.
mine.
All right, Andy, what's your
Well, I
limits?
think the question's too easy in the sense that, yes, I don't think we should be able to use genome editing to intentionally cause harm or mass human genocide. So there's one limit. But I think if we're going to take baby steps, let's start with the legislation and the regulation that we have, which is the HFE Act. Look at how we currently regulate selection and build upon that if we were ever to consider using genome editing. But if we do it, we're going to have to change the law, and that will only happen if you guys vote for a parliament that changes the law.
Alison, what should the limits be?
I'm going to stick to my field of IVF because I think really we should be allowing just about anything in terms of gene editing on the early human embryo that's not going to get put back into a human being to make a baby, but will give us a huge potential benefit in understanding the IVF process, to improve the IVF process so we have more successful treatment.
So only for research, not for
I said I'm going to
embryos.
just cut on stick to that, but yeah, definitely for research. So
yep.
We need to change the act there.
Chris, what should the limits be?
So I think we need quite tight limits initially because I think there's so much research potential and medical potential that if we don't have limits on it and people are scared by it, we risk that important research not being conducted. But in principle, as technology gets more and more advanced, we learn more about genetics, I think we should just use the technology as we can to make the world a better place, and that's a discussion we need to make. What does it mean to make the world a better place?
All right, on that grand note, please join me in thanking the panellists.